Okay, so we are going to go over chapter 8 today. Let me get that screen. I don't think that this will take up the whole hour and a half, so hopefully you'll be done a little bit early today. Okay, so chapter 8, biodiversity, microscopic organisms, a little bit about evolution, and we're just gonna go ahead. Sorry about the computer, I'm trying to get this situated. Okay. Let's get started. Okay, so a little bit about backstory. So it's important to remember that evolution is a theory. We still have to hypothesize about it because no one can go back and test and observe what happened millions of years ago. But we do have fossil records, which tend to show an increased level of complexity over time. So that's how we can kind of extrapolate back how old something may be and what may have come first. So we know from the fossil record and the rocks that we have that the early Earth was not very hospitable. We will never know for certain because we can't go back in time unless we get a time machine at some point in history. We can't go back in time now and see how life originated, so we have to hypothesize what might have occurred. So it is important to remember that it is a hypothesis, that the stuff that we're going to talk about next. And I will point out a few things as we go along. So, questions. How could the first living cells actually arise? How could that have happened? So one hypothesis about this is through biogenesis, which is the formation of new organisms out of non-living matter. So using what you know from a typical animal or plant cell, how do you think the first cell resembled your own cells? Was it similar? Was it different? What may have been different? Okay, so a pretty famous experiment done in 1953 by Urey and Miller demonstrated that it is possible to get organic monomers, or using organic monomers, you can form simple organic molecules. So, for example, like nucleotides and amino acids. So like adenine and thymine and guanine and cytosine. So they did this experiment. You can see it here on the slide where they used water, they heated it up, and in this container here, they had gases that they felt were present in the early atmosphere. And then they had electrodes that go into it to stimulate lightning. And with the hot water vapor and those inorganic, well, no, some of them are organic, and those molecules with the stimulation of electricity, and then you condense it with cold water, you get very simple organic monomers, so single small organic molecules. So that's a hypothesis of what it could have, how it could have happened. A word of caution, don't take this as this is fact, this is how it occurred. We have to remember this is a hypothesis that was an experiment done in controlled settings in a lab, and we know that our environment is not as controlled as you can have in a lab. So just to keep that in mind. Okay, so life does depend on a molecule that can copy itself and pass down information from one generation to the next. We have just spent some time talking about DNA. And so for most life, DNA is that molecule. However, RNA may have been the first duplicating molecule because it is simpler, it is a single strand. And we find that simpler life forms or organisms use RNA, like viruses, for example. Cell membranes may have formed spontaneously, and this is very similar. So if you drop oil into water, it balls up around itself. The oil repels water. Well, that's the same way that phospholipids repel water, and they form that double layer membrane. It's hypothesized that the early cell membranes were formed very easily as these lipid molecules came in contact with water. They repelled water and attracted to each other. Okay, prokaryotes were the first group of life to live on Earth and to evolve. So prokaryotes have very simple, smaller cells compared to eukaryotes. They're smaller, they do not have a true nucleus. They are more simple, they have less complex structures. So looking at the fossil record, we know that prokaryotes seem to have originated first and were the only living organisms for a long time. Today, we can find prokaryotes everywhere. Anywhere there's life, and sometimes when there isn't other life, you will find prokaryotes. So this picture down here is of the Grand Prismatic Spring at Yellowstone, and you have organisms, prokaryotes, that are living in that hot spring and help give it those colors as well. I'm hoping to go to Yellowstone in a couple weeks. Cross fingers. Okay, 2 domains of life make up prokaryotes: bacteria and archaea. So remember, there are 3 domains. We have bacteria, archaea, and animalia. Yes, I said that right. Okay, so in the domain bacteria, they are all single-celled organisms. You find them everywhere, and some cause disease, some are beneficial, and some are kind of neutral. So we'll talk about that later on in this lecture. The domain archaea, also unicellular, and you find these in extreme environmental conditions. So looking at some unique cellular structures and properties, nearly all prokaryotes have a cell wall. So not just a plasma membrane, but a cell wall. And this gives it protection in a wide range of environments, and we will talk about those different types of environments as we continue on in this chapter. Many species are mobile, so they'll have flagella or they'll have cilia, so they can move throughout a medium. Usually it's water. So flagella propels the cells. Many prokaryotes also have a sticky capsule on the outside of the cell wall. And this sticky capsule provides protection. It makes it hard for things to penetrate through the cell wall and the capsule of a prokaryote, and it makes it easier for that prokaryote to stick to host cells, which makes it easier for us to get infected. If let's say we breathe in a bacterium, it has that sticky coating, it's going to stick to our cells in our trachea or our lungs much easier than if it didn't have a coating. It sticks, then it's going to be able to infect. Okay, so prokaryotes have some unique cellular structures and properties. Some have the ability to form an endospore, and this is a thick protective coating that protects that prokaryote from harsh environments. It's like a miniature spore. It protects it from the environment until it's more hospitable, then it comes back to life, essentially. Prokaryotes reproduce by splitting in half, which is called binary fission. I am sorry for this computer. I'm trying to get it. Okay, I'm not going to touch it. Okay, so prokaryotes reproduce by binary fission, which is the simple squeezing two cells in half, and now you have squeezing one cell in half, and now you have two cells that are identical. Okay, this type of reproduction can end, or not end, but result in a very large population over a short period of time. Bacteria can replicate about every 20 minutes, maybe faster depending on the bacterium. And if every cell in a culture is reproducing by binary fission every 20 minutes or so, that initial population gets very large very quickly. If you ever have the chance, one class that's really kind of, I think is fun and really interesting is microbiology. So if you ever get the chance, I would highly recommend taking it. You learn a lot of really cool stuff, and you go in more depth about bacteria and viruses. We're just going to touch the surface. Okay, so looking at bacteria, there are different shapes of bacterium, different structures. We're going to talk about three basic shapes. The cocci, these are cells that are circular in shape, and they can be found alone, or they can grow in colonies and into chains or clusters. Bacilli, these are rod-shaped bacterium, and these, again, you find them by themselves or in chains. So if you had a chain of bacilli, it would look like that. And then spiral-shaped bacterium. These can be curved or spiral-shaped, and these ones you find by themselves. A really good example of a spiral-shaped bacterium is the one that causes Lyme disease. It is a spirochete, which means it's spiraled. Okay, different nutritional habits. They eat different things or consume different things. They don't actually eat because they don't have mouths and intestines, but they engulf and break down different substances. So in addition to being able to carry out photosynthesis for prokaryotes, many of them do carry out photosynthesis. Some can produce their own food directly from the environment. So not from the sun, but from the environment. One really good example would be an organism that lives in the deep sea thermal vents. This is an example here. And they can get nutrition from that sulfur-rich superheated water that comes from that hydrothermal vent. And this is deep, deep in the ocean where there's not a lot of, no, there's life deep in the ocean, but we don't know very much about it because it's hard for us to get down there. Okay, prokaryotes can also produce biofilms, and these are colonies that attach to surfaces like rocks or living tissues. So a great example of a biofilm is plaque on your teeth. So if you remember when you were a kid and the dentist came to school, or you did it at the dentist's office and you chewed one of those little plaque tabs that make the plaque, it dyes the plaque so you can see where you need to brush better. That just basically shows you where those colonies are on your teeth, where that plaque is developed, which is a biofilm. Okay, so you often find prokaryotes that are in the domain Archaea in extreme habitats. So some examples, methanogens, these are Archaea that produce methane gas. Halophiles, like salt lovers. Thermophiles can live in extreme heat conditions. So one example of where you have halophiles, salt-loving archaea, is in a lake in Africa called Lake Natron. And it has this orange color. It's due to pigments found in the salt-loving, these halophile archaea. So methanogens produce methane, and that's their waste product. You can find them in thick mud in the bottom of swamps or bogs, or you can find them in landfills. So there's vents in landfills that help vent the methane gas that's from below from the breakdown of all of the trash. If you weren't able to vent that methane gas, you run the risk of explosions and fires. So that's why they have vents in landfills. Okay, halophiles, those are salt lovers. Ooh, side note, going to Salt Lake next week. Is it next week? Oh my goodness, it's next week. After class is done, going to be driving through Salt Lake City, and I'm going to stop at the Salt Lake so my son can swim in it, because it is a trip to swim in the Salt Lake, because you basically float on top because the concentration of salt is so high and there's salt crystals on the bottom. It's so cool. It's an experience that you're not going to get hardly anywhere else. But I digress. So halophiles, salt-loving archaea, can live in salt concentrations up to 5 to 10 times higher than saltwater, seawater in the oceans, which is pretty salty. So a couple places where you would find halophiles, the salt flats in Bolivia or the Dead Sea in Israel, two great places where you would find halophiles. Sure, you'd probably find them in the Salt Lake as in Utah as well. I'm hoping so. Okay, thermophiles can tolerate high temperature environments. They are the heat lovers, and they can tolerate temperatures above boiling. And it is thought that these conditions are similar to those faced by early organisms a long time ago. So this is that same picture I showed you before of the Grand Prismatic Spring in Yellowstone, which, cross my fingers, I hope we get to stop by there as well on our way back home, because we're going on a trip soon. Get out of Lincoln for a bit. Okay. Prokaryotes in the domain bacteria are very common and very numerous. So the ones that we've discussed were in archaea. So bacteria, they can thrive in almost any habitat, including in and on your body. And there are a lot of beneficial bacteria that live in your body and on your body as well. Yes, there can be some harmful ones, but those are kind of, in the grand scheme of things, those are almost the exception to the rule. Most bacteria is not harmful and can make you sick, but it is there. So bacteria, as I said, can be harmful or it can be helpful. And each bacterium is singular, bacteria is plural. So even though they're very small, their collective impact can be very large. So when large colonies together can make quite an impact. Okay, helpful bacteria. So we're going to talk about some examples of helpful bacteria. Bacteria can be nitrogen fixers. And so they convert the nitrogen gas from the atmosphere in the soil, and they convert it to a form that can actually be used by plants. Then it's taken up by the root system and incorporated into the plant. Bacteria are also important decomposers. And without the breakdown of dead organisms, plants and animals, we would not be able to survive. Everything from the beginning of time would still be here because it didn't decompose, because bacteria has such a major impact on that. So we couldn't survive without bacteria and its important role as a decomposer. How can it help our society? Bacteria is used in sewage treatment plants. And so these microbes decompose that lovely sludge and recycles nutrients and makes it into a safe form again that can be then disposed of. Or there are many cities around the country who use treated wastewater for watering plants and yards. Maybe not yards, but I used to live in California. That's where I grew up. and all of the grass or the plants that are on the sides of the freeways, it had little signs that are up there saying it was treated with wastewater, treated wastewater. So it was considered safe, but it was used, the treated waste, the water that was purified from it, to water those plants, because most people are not going to want to use that water for anything else. Okay, bacteria is also important in bioremediation, and they can be used to remove pollutants from the environment. And so this picture here, they are spraying bacteria here, oh, that's a horrible arrow, to help with an oil spill. Great example of bioremediation. Pathogens are bacteria that is harmful, that can cause disease. And as I mentioned before, relatively speaking, few bacteria actually do cause disease. Most are helpful or neutral to us in most ways. But some can cause illnesses. So here we have Staph aureus or Staphylococcus aureus, which can cause, I think MRSA is, no, MRSA's staph. I'm not 100% sure. I'm a little rough on if MRSA is staph or strep. I think it's Staphylococcus. So MRSA is methicillin-resistant Staphylococcus aureus. That's what MRSA stands for, MRSA, which is a very difficult infection to treat. Bacillus anthracis, that's anthrax. Salmonella, and then here's Lyme disease caused by a spirochete, a spiral-shaped bacterium. Okay, how do bacteria reproduce? They reproduce by binary fission, so basically splitting into two and creating two new cells that are identical to each other from that original parent cell. So as a result, because binary fission occurs and everybody's, well, the cells that are splitting in half are identical to each other, you end up with colonies that are all genetically identical. But this does not mean that bacteria doesn't have ways to recombine genes and get variation. So we've talked about recombination with human chromosomes. Bacteria does recombination as well. So we're going to talk about some ways that bacteria increase their diversity. So we know that they reproduce asexually. They divide in half by binary fission, but there are mechanisms to increase diversity and transferring genetic information from one bacterium to another. So there are four processes that we'll talk about briefly: transformation, transduction, conjugation, and plasmid transfer. So first is transformation. These are pieces of DNA that can be taken up by other bacteria and integrated into their bacterial chromosome. So here we have an example. We have this foreign little segments of DNA. It's taken up by the bacteria cell into its chromosome. Note that the chromosome is circular. And through that process of transformation, it inserts those fragments of DNA into the bacterial chromosome. That is transformation. Once it's incorporated into that chromosome, then it can express that foreign DNA, whatever that DNA may code for. Okay, a bacteriophage is a virus that infects bacteria. So a bacteriophage is used in transduction, and it transfers DNA from a virus to bacterium. So we have this bacteriophage, this virus that infects bacterium. Inside is its DNA. It inserts its DNA into the bacterial cell. It causes the bacterial cell to become basically a manufacturing plant for new viruses. And it breaks down the bacterial chromosome it's actual that bacteriophage DNA is used to create new virus components. It builds new viruses, and then the virus bursts free from the bacterial cell, killing it. So these viruses kill the bacteria cell. They take over the cell's machinery to create hundreds of copies of viruses, which then, when they burst free, they kill the bacteria cell, and then they go on to infect more. Okay, another way that you can get DNA or get variety is through an actual bridge between two bacteria cells. So they use this hollow tube, essentially, that's called a pilus or a sex pilus or sex pili if it's plural, and it forms a physical connection between two cells. And then it can transfer that segment of DNA from one cell to another through that hollow tube. So this happens through conjugation. This is when a donor cell transfers a chromosome copy through that mating bridge or the sex pili to the new cell. So here we can see here, there's our mating bridge transferring chromosome copy to the next bacterium through that physical hollow tube. Okay, and then the fourth one is plasmids. A plasmid is small circular DNA molecule that lives in bacterium. It's not the bacterial chromosome. It's a much smaller little circular DNA. So plasmids have the ability to replicate independently of the actual bacterial chromosome, and that allows it to replicate and pass on that plasmid to a new bacterial cell, have it take it up into the new cell. So you can see down here, here are the little, there's two plasmids there. One is transferred and taken into the new bacterial cell where it can be expressed and replicated independently of the much larger bacterial chromosome. Okay, the endomembrane system is unique to eukaryotic cells. So I'll show you what we mean by endomembrane system. It's made up of two main components. Internal membranes, like the rough and smooth endoplasmic reticulum and the Golgi apparatus, those are internal membranes that are interconnected. And then membrane-bound or membrane-enclosed organelles like mitochondria and chloroplasts are also part of that endomembrane system. So internal membranes likely evolved from the plasma membrane folding inwards from an original cell. And so this picture here shows, okay, we have a hypothetical ancestral prokaryotic cell, and at some point in time, the membrane folded in on itself. And that membrane folding in on itself may have caused or led to that endomembrane system, the endoplasmic reticulum, the Golgi apparatus, the membrane around the nucleus. All this is hypothesized to have formed by this enfolding of the plasma membrane. Again, this is a hypothesis because we don't actually know how it happened because we weren't there. So all we can do is hypothesize. Okay, along with that is the formation of the mitochondria and the chloroplast. This likely occurred by endosymbiosis. So symbiosis is a term that refers to a relationship between two organisms. Endo means in or inside. So this means that we basically have organisms surviving inside another in a symbiotic relationship. So what is thought to have occurred is we first would have to have a cell form that endomembrane system first. Then after that, at some point in time, it is believed that a bacterium that metabolizes oxygen was taken into the cell, which formed that double membrane, which eventually became mitochondria. Then later on in time, the cell or its daughter cells engulfed a photosynthetic prokaryote, which as an engulfed, we had a second membrane form around it, which eventually became chloroplasts. So that is how it is believed that the endomembrane system was formed and how mitochondria and chloroplasts came to be inside cells, that they were originally prokaryotes that were independent of that cell. Okay, talk about protists. They are very diverse. So the term protist is used to describe all eukaryotes that don't belong to plant, animal, or fungus kingdoms. So a little bit about protists. Believe that they evolved about 2 million years ago. 2 billion years ago. This is based off of what we see from the fossil record. We do see single-celled organisms in the fossil record. Most protists are going to be single-celled organisms. Some will be multicellular. We'll talk about one major example of that as we continue on. Actually, a couple examples of that. They are all eukaryotes. So protists are eukaryotes who have that membrane-bound organelle. They're larger and more complex. Okay, so protozoans, they obtain nutrients by eating. So an example would be Giardia, which is an intestinal parasite you get from contaminated water. This is one reason why you don't drink untreated water. If you're in a creek or near a lake or a river, don't drink that water because you don't know what was upstream of it. And it could very well have contained Giardia. I was unfortunate enough once to get Giardia on a trip to Belize in Guatemala. I think I got it from cliff diving into this really cool pond. Really cool deep pool that was formed by a river, and I think some water went up my nose and down my throat, and I got Giardia. So you got to be careful about that. Plasmodium is an infectious protist that causes malaria. I've not had malaria, thankfully. Whenever you travel somewhere where malaria is common, you take a prophylaxis, which basically helps prevent you from getting sick from malaria. It kills the malaria protist before it can really make you sick. And then Paramecium, single-celled organism that has the cilia that beats back and forth like rows on a canoe. And those help it propel through aqueous environments. So protists are very diverse. So one example of a protist is an amoeba, and these are single-celled organisms. They have a lot of flexibility, and they can have extensions that move out, and they can you can actually see them move. It's really cool. If you take microbiology, for example, or you may, and when you're able to take a lab that's in person, if you're lucky, you may be able to see amoeba under the microscope to see them move, and it's really cool. Slime molds, another kind of interesting organism. They look like fungus most of the time, and they grow and live a lot like fungus as well, off of dead, decaying matter. Algae, or singular alga, people usually say algae. These are photosynthetic protists that produce their own food by sunlight through photosynthesis. So algae can be single-celled or multicellular, like this large brown kelp. And those can be hundreds of feet long. They're complex protists. Okay, So, question for you to think about, and this might be our attendance question for today, is what makes multicellularity special? What do you see as major advantages and disadvantages of a multicellular organism versus a unicellular organism? Is it better to be a single cell or multicellular? Pros and cons. So I might make that our attendance question. I want you guys to think about that. Okay, origin of multicellular life. A whole nother step of complexity. So this again is a hypothesis because we don't know what truly happened because no one was there. So we hypothesize that it happened through a series of steps where you had a single-celled organism that developed into a colony of likes or similar type cells. And then over time, that colony was able to have cells that differentiated, cells that had different functions. So cells for eating, cells for movement, and then even later cells for replication or reproduction. And so we believe that it occurred in a stepwise fashion of complexity. So this we're just going to go through real quick. So for almost 2 billion years on Earth, life was unicellular, made up of prokaryotes and eukaryotes. And relatively short period of time ago, so about 1.25 or 1.2 million years ago, multicellular life occurred, and then complexity increased rather quickly. And latest on the scene was humans. We are believed to be one of the last or the latest evolutionary steps. Okay, viruses. This is particularly relevant to us today as we're dealing with the coronavirus. So the question, we've asked this before, is a virus alive? And that was the very first chapter. William, do you remember? Is a virus alive?
No.
Do you remember any reasons why?
I don't remember.
That's okay. That's totally okay, because I'll tell you. Okay, so some of the characteristics of viruses, and then we'll talk about a few that are not there. Can viruses reproduce? They are not able to reproduce on their own. They have to use a host cell to reproduce. So they can't reproduce on their own, which is one of those characteristics of life. Growth and development, they don't show growth and development. They don't have energy usage. They do show order. They do show order. I have a picture coming up to show you. They have kind of a beautiful complexity to them. They are not made of cells. So those are so they cannot reproduce on their own. So that's a no. They don't show growth and development. Why is it erasing? They don't show energy use. I would say they show order based on their overall structure, and they are not made of cells. Okay, so here is a drawing of what a virus looks like. It has no cells. It cannot reproduce on its own, which are two of those defining characteristics. It has to infect a host cell in order to reproduce. It takes over the host cell machinery to replicate viruses, and then it kills that host cell as it releases those viruses. So here we go. Bigger picture of what a typical virus looks like. It has these recognition spikes on the outside that help the virus to recognize certain types of cells, and certain viruses only affect particular types of cells. So for example, the coronavirus seems to target respiratory system. It does target other systems as well, but one of the major areas that is infected or affected is the respiratory system. And so it recognizes those particular cells and attaches to them and then inserts the viral DNA into there, and it takes over that cell. Viruses do not have a nucleus. They do have genetic information inside there. It can be DNA or RNA, which can be double-stranded or single-stranded. So there's a lot of variety of the genetic information of what you can find in a virus. And then the capsid, made of these little balls or bubbles, which is the outer coat of the virus. And it's made of proteins, and it's found in a regular pattern, which shows a little bit of complexity, but it's not a cell. Okay. So a little bit more about a bacteriophage. Remember, this is a virus that infects a bacterium. So the virus can enter the bacterium in one of two life cycles, either the lytic cycle or the lysogenic cycle. In the lytic cycle, remember, lytic refers to lyse, which means to burst or break. So this is a cycle where the virus is using the cell's machinery to replicate more viruses and then is released and kills the host cell. The lysogenic cycle is where that viral DNA is inserted into the bacterial chromosome, and it stays there and it's replicated over time. And so you get a whole colony that have this viral DNA inserted into its chromosome. And it can be dormant for long periods of time until eventually it will re-enter that lytic cycle. So lytic cycle is kind of, I would almost say it's the active cycle, where you're actively making more viruses and you kill the host cell. And the lysogenic is kind of the dormant phase, where the virus is being replicated and it's dormant and it's not harming the bacterium until it enters back into the lytic cycle. Okay, viruses can infect a broad variety of plants and animals. So one virus, this one here, is of the influenza virus. We had a very big Spanish flu outbreak in 1918, which people are comparing what's happening now globally with the coronavirus to what happened with the Spanish flu back in 1918. This is probably the biggest pandemic we've had in 100 years, from what I've read. So don't quote me, I could be slightly off on that, but it's being compared to what we had to deal with in 1918 with the first, the influenza virus. Okay, showing infection of plants. One example, this one is the tobacco mosaic virus, which, if you see, this is an electron micrograph, this picture here that's in black and white. You can see the proteins, those little bubble dots, and it's regular. It has order and organization. Viruses can look kind of pretty under the microscope, but they do, and this one here causes that mosaic virus. Okay, another virus that is relevant to us humans is HIV, the human immunodeficiency virus, and it attacks the immune system. So the disease AIDS, the acquired immunodeficiency syndrome, is caused by HIV. And HIV is kind of special because the virus is called a retrovirus. Its genome is made of RNA, and it targets our immune system. We know that it spreads by sexual contact or in blood products. So one good reason to always use protection and not to do drugs, to avoid contracting HIV. I will tell you, my grandfather had HIV. He was a hemophiliac. We talked about the hemophilia, but he was a hemophiliac and had a bad blood transfusion in the 1970s. And he passed away in mid 1980s. I think he was 85 from AIDS. And that was when people were really starting to figure out what AIDS was and what a horrible disease it was. So it is relevant in my personal history because of my grandfather. HIV relies on an enzyme to be able to replicate. So reproducing, it has to use the enzyme reverse transcriptase. And this converts that viral RNA into DNA. So it's going backwards. That's why it's called reverse transcriptase, because typically we copy DNA into RNA. But if we start with RNA, we have to have a way to make DNA. So that enzyme reverse transcriptase is the key to that. So during normal DNA expression, the host cell will make new viral RNA. And essentially, when this HIV virus comes in contact with the host, it infects the host cell, takes over the host cell machinery, just like we talked about in the lytic cycle, and makes more HIV viruses. And because it's taken into a cell, because it also is affecting the immune system, makes it difficult or impossible to recognize an infected cell and helps kill white blood cells. So here we have kind of a life cycle of HIV starting up here at the top. So HIV binds to a human cell and it injects its viral DNA. It uses that enzyme reverse transcriptase to make a double-stranded viral DNA molecule. So that reverse transcriptase is really important. Once you've made that double-stranded DNA molecule, you insert it, or it is inserted, into the host's DNA. That host DNA then is able, through transcription, to transcribe lots of copies of viral RNA, which then goes out to the cytoplasm and more viruses are made. A new virus cell is assembled and then exits that host cell. In this example, it doesn't necessarily kill the host cell right away, but over time it kills the host cells and it infects white blood cells. And so it infects and then kills white blood cells, which affects your ability to fight off infections, which when enough white blood cells are killed, you enter into the acquired immunodeficiency syndrome stage, where infections that normally you wouldn't get sick from because you can fight them off, you'll get sick from these opportunistic infections and can lead to you passing away. Like pneumonia is one of the common diseases that occurs when someone has reached AIDS, the AIDS stage of HIV. Okay, so question, can a virus evolve? If we've learned anything in the last, oh my goodness, 6 months now since coronavirus has come about, since we've kind of realized it. Actually, I think they first discovered it in December, but it didn't become widespread knowledge until, I want to say, January. We know that this virus had evolved. It is a new novel strain. Viruses can and do evolve. They can undergo natural selection. Okay, talking about what a pathogen is, this is anything that can cause disease. So living organisms like bacteria, fungi, and protists can all cause disease. Non-living viruses can also be pathogens because they can cause disease as well. So 2 other pathogens that are smaller and even simpler than viruses, prions and viroids. So we'll briefly talk about those and then we'll be done with our lecture for today. So we'll be done a half hour early today. Okay. So a prion, this is an infectious protein. And it is essentially a misshaped protein that would normally occur in the human brain. And this misshapen protein cluster together and cause dysfunction in the brain and leads to death. This is also known as mad cow disease. The Creutzfeldt-Jakob disease is also known as mad cow disease, which is really big when I was younger, but it's not a huge area of concern, at least in the US anymore. But for a while, it was a big problem, especially in Europe. Viroids, these are small circular RNA molecules that are able to infect plants. So where prions are infectious proteins that affect human brains or the central nervous system, viroids are RNA molecules that infect plants. They don't code for proteins, but they use the plant cell machinery to replicate and make their own enzymes. So an example of a viroid is a potato spindle tuber viroid, which causes the cracking you can sometimes see in potatoes, especially if they're organic. You'll see these viroid, you'll tell if they're infected by viroids by the cracks in the potato. So next time you look at a potato or you go to the store, if you see a crack, now you'll know if you crack on the potato that it's caused by a viroid that's actually infectious. And that is the end of our lecture today. Looking forward, I'm gonna pull up Canvas just really quick. Maybe. Hello. There we go. Pulling up Canvas really quick, kind of giving you a heads up about what's happening in the next week. We will be finishing our class on June 30. In this next week, you have an exam on Wednesday, June 25. I think that's Wednesday. Oh, I'm sorry. The 25th is a Thursday. So our exam is on Thursday, and we will lecture on chapter 9 Thursday morning, and then do a quick review. If there's questions, we'll cover those. And then we'll have another lecture on Friday to begin our last section. So just keeping in mind that Thursday is when all of your homework is due. You have 2 quizzes, quiz 6 and quiz 7. One quiz is due today. So Monday by midnight, you have to have quiz 6 done, which is over what we just talked about. And if you have lab, you have 3 labs, I'm sorry, you have 2 labs that need to be completed by Thursday. Any questions? You're good? Okay. All right, look for your attendance question in a little bit. And if you have questions between now and our next lecture, which is Thursday, please email me or text me, and I'll get back to you as soon as possible. If you don't hear from me, text me again, and I'll get back to you, okay? Have a fantastic day, and I will talk to you soon.